Well, thanks everybody for joining us today. I'm Tim Nelson. I'm the director of the Todd and Karen Wanick Family Program for Hypoplastic Left Heart and the director of the HLHS Consortium here in Rochester, Minnesota, at the home of Mayo Clinic, with our program was launched. And with us today, we have our biomanufacturing cell production team at HeartWorks. And we have many people from all over the country today joining us. So thanks for joining us. We're excited to give you an inside tour of our research and development, product development infrastructure here, and share with you how we're developing new therapies to treat congenital heart disease. So we're going to have plenty of time to have questions, and I encourage you all to either chat and type your question out. Cal will be watching for that. But more importantly, it's always fun just to interact with all of you, to take yourselves off mute and we'll have a conversation. So our goal is to walk through a tour and give you context of how we develop products for congenital heart patients and our clinical trials. And we'll take about a half hour to do that. And then we'll have a time for you all to ask questions and engage with us and interact with the team that's doing this work. So that's kind of our game plan for this morning. So again, thanks for joining us. So as a way of introduction, I thought I would just spend a few minutes to build the context of where we've come from over the last 10 years. So with the investment of Todd and Karen Wanick and their family, they've created a program focused on hypoplastic left heart with the goal of transforming the way we take care of single ventricle kids. And we've been doing this for about 10 years now at Mayo Clinic, and we've developed an ecosystem of partners from basic science research all the way to clinical trials. And we'll talk about all of that today. The important part is it's a team of teams approach across the country with many, many people working together to make this happen. And we're grateful for all those partners. The most important partner in all of this is HeartWorks. And that's where all of this funnels through with our biomanufacturing facility. And that's going to be the focus of today's tour, is how all of the research funnels into a biomanufacturing facility. And then out of that, we create clinical trials where we're treating kids across the country. So HeartWorks becomes the hub of how all of that product development flows. And you're going to get an inside view on some of those products of how we work collaboratively to develop this. So as I'm standing here in our basic research lab, this is what we started out with about 10 years ago, with genetics, stem cells, preclinical models, and the hope and the dream of creating clinical trials to treat congenital heart patients with cell-based therapies. And it started in a lab discovering what we could do to make this dream a reality. To do that, we typically did research in small scale plates like this, where we would discover things. And I built a lot of hearts in plates like this to cure mouse hearts of heart disease. And we spent a lot of time throughout my career as a scientist doing things in preclinical models with the hope that we could learn something in small scale academic research that we could think about, dream about the day someday where we could actually treat patients. And that's what a lot of academic labs do. They write grants, we get papers, and we focus on new knowledge of what we could hope to be possible. But as we started doing that and got more proficient at it, we realized that we needed to go to a scope and scale like this. And we needed to expand the stem cell research that we're doing to be able to grow at much larger volumes. And what you're looking at right here is a jar that you'll see in a little bit more of. But inside that red ball there is actually the size of a human heart of when a baby is born. And that's the size of the heart muscle that we need to create bioengineered products to be able to treat patients. So going from a small scale like this to a large scale like this has really been the focus of the last 10 years with our manufacturing partner now at HeartWorks. Let me set the stage for this a little bit as we go into the lab. What we started out 10 years ago dreaming about being able to do was to take a piece of your skin, to collect it, grow it, retrain it into stem cells, and allow that stem cell then to create your heart muscle. Now that sounds a little bit science fiction, a little bit creepy, but we're going to show you today that this is a reality of what we're doing. So taking a piece of skin, growing it into stem cells, and then teaching it to become your heart, beating, contracting heart muscle. And I hope by the end of the tour here in the next 20, 30 minutes, you all are going to see for the first time a human heart muscle beating in the lab that we've bioengineered from somebody's piece of skin. So that idea in an academic lab is really just an idea and never goes anywhere. And you have to have partners to be able to bring that through the research and development into clinical trials. We looked for those partners. Those partners did not exist in the congenital heart world. It's too small of a market. There's not big companies involved. And we didn't have those partners that we could partner with to bring the research out of the lab, like here at Mayo Clinic, into clinical trials. And so with the team over the last 10 years that now is at HeartWorks, we've built that infrastructure through this collaborative partnership. And we've co-developed this. And so as you see where I'm standing right now, I'm going to turn the camera and you're going to see right next to our research lab is a state-of-the-art biomanufacturing facility. And we're going to take a tour inside here to Casey, who you'll see through the window here standing right in there. He's also in the cell manufacturing lab. So he's literally right inside this plexiglass facility from where I'm standing in our research lab. And it's these professionals and these experts that scale up the production of our stem cell manufacturing that allows us to get at a scope and scale that we can bring it to clinical trials and to patients. So I'm going to hand it over to Casey, and he's going to talk about what he and his team do inside this biomanufacturing facility to make heart muscles at a scope and scale that we can use for clinical trials. Casey, do you hear us?
Yeah, can you hear me?
You bet.
All right, thanks, Tim. And thanks to all of you for joining us today. We spend a lot of time taking care of these cells, and they don't necessarily talk back to us. So it's really awesome to share what we do with some engaged people, and hopefully you have some questions later on. So as Tim mentioned, this whole process takes about 6 to 7 months. It's a long, grueling process. And one of the most important tools that we have is this clean room that Tim mentioned. And how it works is it's called positive pressure. So above me, there are HEPA filters that are pushing air into this clean room. And that creates a positive pressure, which makes the air move out of the clean room. So nothing can infiltrate and get into the clean room, if that makes sense. Then within these rooms, we have these biosafety cabinets, which operate in the same manner. So anytime that we're touching these cells as they grow and differentiate, they're always in this ultra safe environment. And then we have our gowns, our gloves. We spray ourselves down with alcohol a lot to kill any chance of bacteria contaminating and infiltrating the product. So as Tim mentioned, it's a 7-month process, and it starts with the skin biopsy. So I think we're gonna have Doug zoom down here. So when we obtain this skin biopsy from the patient, it's about as large as that little red dot in the plate that I'm pointing to here. And over the course of about a month, we cut that into little tiny pieces, and slowly but surely, these adherent cells start to grip onto the plate, and they start to replicate and grow bigger and bigger and bigger. Once we have enough of these cells, we actually infect them with a virus that allows these cells, which think they're skin cells, to kind of be reprogrammed into this stem cell state. And if you're not familiar with stem cells, these are kind of the cells in the beginning of development that can turn into multiple different types of cells. So after about another month, we start to pick these pure stem cells and take care of them. It's kind of like cleaning a weeding garden. We want this perfect pure sample, so we have to get rid of everything that we're not really very interested in. And eventually we'll have just one plate of good pure stem cells. From that point on, the analogy that we use in the lab is kind of like hostas. If anybody has a nice garden at their home, you kind of split these cells apart and then allow them to plant their own ground, and they'll start to grow into more hostas, more hostas, more cells, more cells, and more cells. So slowly we have this logarithmic growth. We start with 1 plate to 2 to 4 to 8, and eventually we have 12 plates of cells. And At this point in time, it's all two-dimensional cells. So think like a pancake on the plate. And once we have enough of them, the growth is kind of unsustainable in this two-dimensional state. So we need to allow them to grow into more like a ball, like a three-dimensional state. And that's when we treat them in a manner that allows them to grow in these flasks. And if you can see, there's pendulums in these flasks, and they spin around and around, and they kind of keep these little balls of cells moving around and around and around. And that allows them to grow even further. And once we expand those cells to a certain point, then we're ready to treat them in a way that sort of pushes them down a cardiac differentiation path. So they start as these stem cells that can kind of become anything. And once we treat them with this protocol, they start to kind of agree to become heart cells. And that's when they eventually start beating. And that's the goal. And it's 7 months long, and our success rate is pretty good. It's not always perfect, but we're learning more and more every day. So now I can show you some pictures of those stages along the. Sorry about this, just getting the camera right. All right, so here's a skin biopsy in the very first stage. There we go. So this is right when we receive the skin biopsy. So this little piece of skin will be cut up into little chunks, and then we'll put a little glass slip over the top of it. And that'll allow it to slowly start to grow these little kind of long fibroblast snake-like cells. And once we get those, they'll start to propagate themselves, and they'll expand. And eventually they'll fill the entire plate. They're like weeds. And then once we have enough of these, like I said, this is when the virus infects these cells and causes them to kind of revert back into this stem cell state. And then eventually we'll start to have these little colonies formed, and these are the pure stem cells that we're looking for. And this is about after 2 months. So this, after 3 months, this is exactly the ideal situation. We have this nice little colony of induced pluripotent stem cells that we can then pick and put on another plate, and then hope to see this more pure growth of induced pluripotent stem cells. So this is kind of the weeding part of the process. There are some good induced pluripotent stem cells here, but there are also cells who just did not want to become stem cells, and they want to become brain cells or anything other than stem cells. So at this point, we have to come in and kind of clean out the bad stuff and make sure that we stick with just the good stuff. And eventually we'll have these pure, nice, good morphological stem cell colonies. So each one of these colonies is made up of a bunch of little individual cells. And then to go back to the hosta analogy, we'll take this, and we actually have a robot that we made that will cut these small colonies into little tiny chunks. So here's a good colony. So this is our homemade robot. We haven't named it, so if anybody has a good idea for a name, that would be. So as you can see, this is the robot cutting these tiny little colonies into even smaller chunks. And what we end up with is this picture. So then these will be scraped off the plate and put onto another one. And each one of these will grow back into one of these. So as you can see, that's how we kind of expand the population of cells.
And just to go back, like Casey, can you go back to that picture of the colony? So after 5 months, when you get this colony, this is really hard for people to fully understand. I just want to emphasize this, that these cells, each one of these cells, you're looking at hundreds of cells now growing in this colony. Those are daughter cells, the hosta analogy, that has grown out of your piece of skin. And you started with a piece of skin biopsy, like the tip of an eraser of a pencil. And now we've reprogrammed them to be induced pluripotent stem cells. So each one of these cells has the magical potential to literally give rise to every cell in our body. From our head to our toe, these cells behave just like embryonic stem cells behaved when you were an embryo that was 7 days old. But these didn't create an embryo. They didn't destroy an embryo. They came from your skin, but they've been trained to think that they're an embryonic stem cell. And that's why we call them induced pluripotent stem cells. And so these cells have the ability to give rise to anything in our body. And that's a really amazing feature that is hard for people to fully grasp, that these cells are your cells, but we now can teach them to become whatever we want. And as Casey and Doug are going to show you now, what we really care about is training those stem cells to become your heart. And that's the tissue that we're most interested in, is your beating cardiac cells from whoever donated that piece of skin. But these cells, as you're looking at them after 5 months, can live in a freezer at low temperatures. We can preserve them for a long, long time, maybe 50 years at least. And we can use these cells to go back to and differentiate many, many tissues. But you're going to specifically see today the goal of making the heart muscle out of these cells. So 5 months is a lot of work, but it's just the beginning of the fun part now.
Yeah, so as I mentioned before, these are all two-dimensional, so just like pancakes. And what we want is more like a basketball. As you can imagine, if you can grow in all 3 dimensions, there's much more room for growth. So once we have enough of the plates in the two-dimensional, we'll treat them in a way that they can survive in these flasks. So instead of those flat colonies, these are little tiny aggregates that are visible to the eye. I mean, very small, smaller than the tip of a pen. When they're swirling around in the flask, it almost looks like little sand going around and around. And this allows us to really produce billions of cells at a time. So these are still in an stem cell state. So as Tim mentioned, we can train them to become whatever we want them to. Our goal is cardiac tissue. So in about a course of 20 days, once we have enough of these cells, we'll try to steer them down the path to cardiac differentiation. And they start to change their morphology and look a little different. And eventually, after about 15 to 20 days of going through this protocol, they'll eventually start actually beating. Hopefully you can see that on the camera. It's really cool to see. It never really gets old. It's always just a good feeling after the 6 or 7 months of work to have it all work out.
Can everybody see those beating masses of balls? They're kind of squeezing and contracting. These are the, Casey, what size are we looking at there roughly?
What size?
Yeah, what size are those balls, to give a perspective? We're looking under a microscope, but what's the actual size?
They're about 600 microns? Or,
so this,
yeah, like a grain of sand or even smaller than that.
Yeah. The thickness of a few human hairs, perhaps, is another way of thinking of it. If you put a couple hairs together and looked on the end of it, that's about the size of what you're looking at under a microscope. So these are small little heart aggregates that are beating and contracting, and you can see that under the microscope.
Here's one more of a up close one, if that.
So it's remarkable to me every time I see these cells that are beating and contracting, is this is a human heart muscle cell that if we put a drug on those cells that increase the adrenaline or the excitement and your heart races, these cells will start contracting faster. If we put drugs onto these little hearts that slow it down, like some people take beta blockers, we can actually cause that heart muscle to start contracting more slowly. So these human heart cells are actually responsive to the same medications that we take to speed up and slow down our heart muscle. They function just like the person that we collected them from, because they're genetically identical to the skin biopsy that they were collected. So it allows us to a lot of things in the lab when we have this scope and scale of what's being produced. I hope you all can see that, because it's always crazy for us to see, but hopefully you can see the beating. It's fun to see.
Yeah, so Our goal is to produce about 1 billion cardiomyocytes, which ends up being about 3 to 4 milliliters. And for reference, a teaspoon is 5 milliliters. And the entire adult human heart is made up to about 2 to 3 billion cardiomyocytes. So, creating the amount of cells that are in a human heart is definitely feasible for us at this point. So as we mentioned multiple times now, it takes about 7 months for that process. We're not entirely sure when we'll be able to do this, and it's something that we're working really hard to figure out, but eventually the goal is that these cells will be incorporated into the heart in a therapeutic manner. And I guess I'll let Tim talk a little bit more about that.
Yeah, so just to recap, in the research lab, we start out with a small scale of plates like this. And now what Casey just showed you is growing it in large vats like this. And in the last picture, Casey just showed you 1 billion cardiomyocytes. That's equivalent to the number of human heart cells that our children are born with on the first day of life, about 1 billion cardiomyocytes. So what Casey just showed you is a pellet of cells that's equivalent to a human heart when they're born, that's being manufactured from a piece of skin. We obviously think that's pretty creepy cool and pretty exciting to go from a small scale to a large scale. And so what are we doing with that? All of you are thinking, how does that matter to me? That's a great science project, but how does this matter? What Casey is doing and what the team in the lab that you see right now where Casey is standing is those cells have now come out of that lab, and with the FDA permission and with guidance from the FDA, we are now testing that in what we call our four-legged patients, our small animals like mice, to test the safety of these cells, but we also are testing these in large animals or primates. And so this is a requirement for the FDA for us to make sure that we can test these cells in safe model systems before we would ever bring them to a patient population. So currently we have many primates that are living with these human heart cells in their hearts to show that it's safe and effective. And we are doing this work over the next, for the end of 2021, and we hope that we will have the safety data that the FDA requires for us to be able to launch the first human clinical trial where those cells will be packaged in a way that we can inject them and deliver them into the heart of a child with congenital heart disease that has a weak heart muscle. Now, one of the key things that we focus and why this is such a remarkable process that Casey has just said, it takes a lot of time and a lot of money to grow this for many, many months. And we've used the word your heart many times here. We're not building heart from a donor that we're then going to use for a bunch of kids. There's problems with that. The problem is that it has an immune reaction. You have to be on immunosuppression drugs to preserve somebody else's cells in your body. But what this team has scaled now for the first time in the world is your cells that are genetically identical to the sample that was started. And so in theory, these cells can be transplanted into your body and they won't be rejected. We've basically rebuilt in the lab your heart so that we can rebuild and re-augment your heart with a surgical technique. Now, the first step of that might be happening in 2022 when we deliver this potentially into our first patient that is in need. But you can also imagine how we can use these cells to grow different parts. Congenital heart defects are a problem of having, I call it a manufacturing defect, if you will, of the heart. There's missing parts in the heart. If we can rebuild those parts, we can cure congenital heart disease by replacing those parts. And it takes a manufacturing facility like this to be able to build it at that scope and scale. So that is one of the exciting areas. I'll pause there, and sometimes people have questions